Let's read. Students, as you might know, it is sometimes difficult to pay full attention at what you are reading. At such times, listening to the audio of that particular chapter can help. As you listen to the audio while paying attention to each and every word, your concentration span improves, thus leading to an effective reading. So, let's start reading together. Standard 9th Science 2, Chapter 8, Useful and Harmful Microbes, page number 88. Useful microorganisms: Lactobacilli, Rhizobium, yeast. Harmful microorganism: Clostridium and others. Can you recall? 1. What is meant by microbes? What are their characteristics? 2. How did you observe microbes? You are familiar with different types of microbes which are all around us but cannot be seen with our eyes. In what way are they related to our everyday life? Useful microorganisms: Lactobacilli. Try this. Smear a drop of fresh buttermilk on a glass slide. Stain it with methylene blue and put a cover slip over it. Observe the smear under the 10x objective of a compound microscope and then with the more powerful 60x objective. Did you notice the blue rod-shaped organisms moving around? They are lactobacilli, a kind of bacteria. They are minute and rectangular in shape. Lactobacilli are anaerobic bacteria, that is, they can produce energy without the use of oxygen. Figure 8.1, Lactobacilli. Can you tell? How is yogurt made from milk? What exactly happens in this process? The lactobacilli converts lactose, the sugar in the milk, into lactic acid. This process is called fermentation. As a result, the pH of milk decreases, causing coagulation of milk proteins. Thus, milk proteins are separated from other constituents of milk. This is what happens when milk changes into yogurt. Yogurt has a specific sour taste due to lactic acid. The low pH destroys harmful microbes present in the milk. Use your brain power. 1. Why do doctors advise you to have yogurt or buttermilk if you have indigestion or abdominal discomfort? 2. Sometimes yogurt becomes bitter and froths up. Why does this happen? 3. Which different milk products are obtained at home by fermentation of the cream from the milk? Do you know what is meant by probiotic yogurt and other foodstuffs that are popular nowadays? Useful microbes like lactobacilli are added to these eatables. Such eatables are healthy because they kill the harmful bacteria like Clostridium in the alimentary canal and help to improve our immunity. Page number 89. Uses of lactobacilli. 1. Various milk products like yogurt, buttermilk, ghee, cheese, shrikhanda, etc. can be obtained by fermentation of milk. 2. Lactobacilli fermentation is useful for large-scale production of cider, cocoa, pickles of vegetables, etc. 3. Lactobacilli and some other useful microbes taken together are used to treat abdominal discomfort. 4. Leavened fodder offered to domestic cattle like cows and buffaloes is fodder fermented with the help of lactobacilli. 5. The lactobacilli fermentation process is used to make wine and some type of bread. Research. 1. How many different industries depend upon the lactobacilli bacteria? 2. Which types of cottage industries and factories can be started in areas with abundant milk production? Rhizobium symbiotic bacteria. Let's try this. Take a plantlet of fenugreek, groundnut or any other bean and sterilize it with a 3 to 5% solution of hydrogen peroxide. Afterwards, keep it in a 70% solution of ethyl alcohol for 4 to 5 minutes. Clean the roots with sterile water and take thin sections of the root nodules. Select good section and place it in a solution of safranin for 2 to 3 minutes. Place the stained section on a glass slide. Cover it with a cover slip and observe it under the compound microscope. The pinkish rod-shaped organisms are the Rhizobium bacteria. Note that we had to search for the root nodules of leguminous plants to obtain these bacteria. Are the Rhizobium bacilli useful to these plants or harmful? Figure 8.2 Root nodules of soya bean plant. Role and importance of Rhizobium. Rhizobia living in root nodules supply nitrates, nitrites and amino acids to that plant and in exchange get energy in the form of carbohydrates from it. Such a mutually beneficial relationship is called symbiosis. Rhizobia produce nitrogenous compounds from atmospheric nitrogen. However, for this process of nitrogen fixation, they need leguminous plants like beans, sweet pea, soya bean, etc. as host. Beans and pulses are rich in proteins due to the nitrogenous compounds made available by Rhizobia. Page number 90. After harvesting a leguminous crop, the leftover roots and other plant parts are deliberately dumped in farm soil to maintain its bacterial population. The use of Rhizobium has helped to reduce the use of chemical fertilizers and thereby their adverse effects. It has also helped to reduce expenses on fertilizers and thus works to the benefit of farmers. Nowadays, seeds are coated with rhizobial solution or powder before sowing. After sowing, Rhizobia enter the plantlets. This is called rhizobial inoculation. This experiment has helped in the supply of nitrogen to cereal and other crops besides leguminous crops. Yeast activity. Bring active dry yeast from the market. Mix a spoonful of yeast, 2 spoonfuls sugar with a sufficient quantity of lukewarm water in a bottle. Fix a colorless transparent balloon on the mouth of that bottle. What changes do you observe after 10 minutes? Mix lime water with the gas accumulated in the balloon. Collect that lime water in a beaker and observe it. What do you notice? Take a drop of the solution from the bottle on a glass slide. Put a cover slip over it and observe it under the compound microscope. Store the solution in the bottle carefully. Do you see the colorless oval cells of yeast on the slide? Some of those cells may have small round bodies attached to them. These are new daughter cells of yeast in the process of formation. The method of asexual reproduction is called budding. Yeast is a heterotrophic fungal microbe that grows on carbon compounds. Yeast is unicellular fungus with 1,500 different species in existence. The yeast cell is a eukaryotic type of cell. Figure 8.3 Yeast cells. In the above experiment, yeast grows and multiplies very quickly due to the carbon compounds in the sugar solution. In the process of obtaining nutrition, yeast cells convert the carbohydrates in that solution into alcohol and carbon dioxide. This process is called fermentation. How is bread made? Find out how to use the solution prepared in the above solution to make bread. Follow the recipe and make the bread. Find out and note down the reasons why the dough rises and makes the bread spongy.